Buenas noches a todos. En algunos minutos empezamos nuestra transmisión con una conferencia especial de David Raeimer sobre la sistemática y evolución de Zoantharia. ¿Sí? En algunos minutos ya empezamos. Hola a todos. Vamos a empezar con la gran conferencia de hoy. ¿Sí? Esta noche tenemos un gran convidado, ¿sí? Invitado, que es David Raeimer. David Raeimer tiene una gran formación. Comenzó su graduación en la Universidad British Columbia, en Canadá, en un curso de ecología. Después hizo su maestría y doctorado en Kagoshima University, en Japón. ¿Sí? Después hice 2 estágios de pós-doutorado, en Instituto de Kuroshio, en Japón, y también en Jamstack, en Japón. Y luego después empezó los trabajos en la Universidad de Ryukyus, en Okinawa, también en Japón. ¿Sí? Desde, uh, desde 2008 está en la universidad y tiene muchos sucesos desde entonces, con más de 32 grants de pesquisa, ¿sí? Con mucha plata obtenida para una serie de diferentes proyectos. También tiene una gran producción de estudiantes, con más de 70 estudiantes de graduación, de posgraduación, en postdoctorados, en estos últimos 10 años de trabajos. Es editor de una serie de jornales muy importantes y ha descrito más de 60 especies de zoantídeos, que los grupos que va a hablar hoy para nosotros, ¿sí? Y como se ve en su flyer de presentación, James va a hablar sobre sistemática y evolución de Zoantharias a partir de ahora, ¿sí? James, please. Can you appear for us?
Yes, so, thank you, Sergio.
Thank
Can
you.
You hear me okay? Everything's okay?
Yeah, it's perfect. Everything's
okay.
perfect.
Great.
Thank you for meeting us to join us this night for us this morning.
Yeah.
For you. It's a real pleasure for us because we are starting our network, Cnidarian Network here in Latin America, and we are trying to make it stronger, like to connect people, to make like a group, in fact, a real network, and of course to bring guys like you is really really important to us. See, so yeah, thank you again and your time.
No, thank you. Just quickly before I start, I'd like to say that I think this is a great opportunity. Online meetings in 2020, I think, are very important for all of us. We can be active with our research. And I will talk a little bit about this, but I think for Cnidarians, Latin America and South America are very important for Cnidarian research. So I hope next year and next year, I hope every year that there's chances for different researchers to meet and talk about Cnidaria. So I think that's great. So thank you very much. And
thank you.
I will, it's morning here, so I'm gonna start my presentation. I've had my coffee, so I'm ready to talk. So let's go to share screen. And I will share the screen here. Okay, so this morning I'm going to talk, or this evening, sorry. Thank you all for coming at nighttime. My name is James Davis Raeimer. I'm at the University of the Ryukyus in Okinawa, Japan. And I'm going to talk for about 45 minutes on Zoantharia. So I hope you have lots of questions. I hope there are some things new that you learn, and I hope you enjoy the talk. It will be very fun to talk to you. So without further ado, let's start. So today I'll give you a quick outline about what I'm going to talk about. Of course, first, you're all Cnidarian experts, I think, but I'll do a very quick introduction about Zoantharia and a little bit about evolution and systematics. Then I'm going to talk about the phylogeny, the molecular phylogeny of the order, and how this has changed over the past 15 or 20 years. Then I'm going to introduce to you three sort of ongoing studies that we have been working on in our lab. So the first one is more higher level taxonomy, families, and genera, looking at systematics and evolution in one suborder of Zoantharia. The next one, I'm going to talk about trying to tease apart very closely related species and understanding how they are related to each other. And then finally, if we have time, I will talk about ecology within species, but I'm not sure if I have enough time to talk about the last part, part C. And then I'll have some conclusions and talk a little bit about the future, or what I think the future is going to look like for Zoantharian research. So let's start with an introduction to Zoantharia. So as you all know, Zoantharia is one order of hexacorals, and I'm going to briefly sort of give you an overview about them from now. So the basic way that I often explain to students in the lab is sort of thinking of them as, and I put it in quotations because we know coral has many meanings, coral without the skeleton. In other words, kind of like a scleractinia without the skeleton. By this I mean they're generally colonial, they're generally attached to the ground, to the substrate, or to another animal. You don't find many that are completely free living and non-attached. They're worldwide. They're in pretty much every marine ecosystem that we've looked for. We have found them, from mangroves to coral reefs, to the Arctic Circle, to Antarctica, to the deep sea, to 5,400 meters. If we look for them, they are there. The current species number in worms is 350. In literature, maybe close to 700. But I think there's probably many more species than these numbers indicate. And we'll talk about that today a little bit. Until recently, they've generally been very poorly studied, very poorly described, and they're not well understood. And so one of the things that myself and some other researchers have started to do is to really try to make people to not forget about the Zoantharia. So let's go a little bit more into detail. So I like to call this their neighbors. I've put up information on 3 orders of hexacorals: Scleractinia, Zoantharia, and Actiniaria. Colonial or solitary. So Scleractinia, except for fungid corals, usually are colonial, at least the ones we see on coral reefs. And I have a picture here of an Acropora colony. Then Zoantharians are also the same. They're colonial, as I said before. Whereas anemones are generally almost always solitary. Even if they're large aggregations, they're colonial aggregations. And so they're not connected by common tissue. The flip side is when you think about skeleton, Scleractinians make calcareous skeleton. Anemones obviously have very strong muscles, whereas Zoantharians, almost all Zoantharian groups, they take up some kind of sand or detritus from the ocean, and they incorporate this into their body wall to make themselves stronger. The easiest way for me, or for anyone, if you go to the ocean and is this a coral, Scleractinian coral? Is this an anemone? If you look at their tentacles, the Zoantharians always have their tentacles in 2 cycles or 2 rows. And they're always in the same location. Anemones may have some coming on the oral disc. Corals have many different arrangements, Scleractinian corals, but Zoantharians always have this 2 rows of tentacles. If you look at the morphology of Zoantharians, they do have a lot of characters. They have a single saphenoglyph. They have usually long and short, or major and minor mesenteries alternating. But the one thing that's very interesting about Zoantharians is they're very uniform in their morphology. Even as far back as Karlgren, I think in the 1920s and 1930s, he was mentioning that it's easy to identify a Zoantharian, but inside Zoantharia, they really the different groups look very, very similar. As well, recent experimental research has shown that a lot of these characters that we look at, for example, sizes or tentacle lengths or oral disc diameter, these things can often vary, especially based on microenvironments. So what you have is a group that's easy to identify to the group, and inside the group, they look very consistently similar, which is quite interesting. The major groupings in Zoantharia, there's 2 suborders currently. And so if you take one of these polyps, and I put some red dotted lines there, if you do a lateral section on a polyp around where the saphenoglyph and actinopharynx is, and you look at their mesenterial format, if you see on the slide, the DD, this is the dorsal directive. Okay, so this is the top directive, and you count the number of mesenteries: 1 incomplete, 2 complete, 3 incomplete, 4 complete. The 5th mesentery in some groups is complete, and we call these macronemina, which is 1 suborder. And in other groups, it's incomplete. So we call this the short ones, the brachynemina. And so these are the 2 major groups of Zoantharians currently, the 2 suborders. Ecologically, these are good suborders. Phylogenetically, I will show you after, they're not. And also recently, we have found 2 species that do not fit this mesenterial form. They're in the wrong, they have the other suborders format, even though they're clearly in 1 suborder. So this is pretty good, but this is not perfect. It's pretty good though. So when you look at Zoantharians, and this is something that I only recently discovered. I was talking to Tim Swain and some students, and basically I realized this. The Japanese name for Zoantharian is sunaginchaku. And sunaginchaku is literally translated into sand wallet. And so I'd been thinking about a Western wallet, you know, with your credit cards and it opens like this. But in Japan, this is what an old wallet looked like. And so it's a bag with a drawstring, like a sphincter muscle. And so Zoantharians, they take up sand in their body wall, but they have this sphincter muscle. So they look like a sack with a sphincter muscle on the top. So to me, sorry, this is a very strange specimen photo. You can see the size there. It's a very large Zoantharian. So this is a traditional Japanese wallet. Put a bunch of sand on there, remove this string right here, and you have a Zoantharian. And in Zoantharia, 9 of the 10 families take up sand and rock to enhance their structure. What this means is histology is very difficult, even though we need histology to look at the mesenterial format or to look at sexual reproduction, for example. Calcium is easy enough to get rid of with acetic acid, but they often take up rock and silica as well. And so to remove this, you need hydrofluoric acid, which can be lethal above 20% concentration. So as you can see from these slides on the bottom, these pictures on the bottom, especially for the groups that take up a lot of sand, morphology is very difficult. And some Zoantharians, in some groups, 60% of their body weight is sand or detritus. So they really are sand wallets. As well, if you want to look at color, there's a lot of interspecific variation in color. This is a picture of one Zoanthus colony in Japan. And you can see 1, 2, 3, at least 4 different morphotypes. You can also see a large variation in the size of the polyps. And so the adult polyps are usually roughly the same size, but the smaller polyps can be quite small. So you have a lot of variation. And this was the state of things until about 1995, when people started to move not beyond, but including morphology, but started to include molecules in their research. And so from there, in the last 15 or 20 years, we've sort of made, several of us have made an effort to really try to understand Zoantharian evolution and systematics. So we've been using museum specimens. Here's some specimens from the Naturalis Biodiversity Center in the Netherlands. We've also been doing a lot of diving, which before coronavirus was fantastic, traveling the world, looking for new specimens or new species, undescribed species. And then also deep sea work as well. And then we've been combining this with molecular methods. Until recently, mainly straight DNA sequencing, Sanger sequencing, but also more recently, next generation sequencing. And making phylogenies. And then with the phylogenies, reconsidering their evolution. And I think in the last 15 years, as a community, we've made a lot of progress in understanding Zoantharian diversity. And so that's what I want to talk about today. So let's talk about the phylogeny of the order Zoantharia. So pre-molecular systematics and taxonomy, the first Zoantharian was described by Ellis from the Caribbean, I think in Jamaica actually, in 1768. Here's a picture, obviously not a digital camera, but you can see it's got a stolon, it's a colonial animal, and this was Zoanthus societus, which is a very common zooxanthalate Zoantharian in the Caribbean and Atlantic Ocean. So it was a bit chaotic in terms of higher level organization until Haddon and Shackleton in 1891. They divided the group into 2 groups, the Macronemina suborder and the Brachynemina suborder, based on the mesenterial arrangement. Species, genera, families, these were erected based on sand incrustation, presence or absence, sphincter muscle shape, host, if they're associated with other animals, et cetera, et cetera. So there was a lot of diversity work done until about 1950 or 1960. A lot of this was done in the 20th century by Karlgren and Pax. And so we had a pretty good understanding about a lot of the different Zoantharians, but the systematics remained with Haddon and Shackleton, 1891, the 2 suborders. But when people started to apply molecular techniques, particularly Burnett in 1995 and 1997, using allozymes, we started to see that there might be some problems. Burnett found that many different species maybe had very similar molecular makeups with allozymes. And so they sort of pointed the finger that we need to start reconsidering phylogeny. And so with that, in 2005, Siniger and colleagues produced the first phylogenetic tree that specifically examined zoantharians. This used 2 mitochondrial markers, 12S and 16S. And basically what they found was it was a basic support for the morphological base systematics of Haddon and Shackleton with 2 suborders, although the phylogeny suggested that maybe one was paraphyletic. So there were suggestions of taxonomic problems of family and genera. And so you can see here, here's the 2 suborders. And so you can see at the bottom, around here, you can see there's a group of Macronemina that's actually branching with the Brachionemina. And so this was something that has been indicated since 2005. Then in 2010, Siniger and colleagues did an updated version with more markers, 16S, CO1, mitochondrial nuclear ITS. They looked at substrate specificity. So what is the zoantharian attached to? Is it attached to a sponge? Is it attached to an antipatharian? Is it attached to the substrate, hard rock? And so they started to erect some new genera, and they split off one of these groups in the bottom here to Hydrozoanthidae, which were the zoantharians associated with hydroids, Dentifrica and such things. And so this was the first sort of important step towards getting a clear understanding of the higher level systematics of Zoantharia. And so in their paper, actually, they have photos of different zoantharians on different hosts, and they suggest that the host association is an important piece of taxonomic information. After this, Fuji and Raeimer, who was a student of mine, and combined with a couple other papers, we found a lot of outlying or weird zoantharians, and this was with increased field work. So we found Abyssoanthus, which is in the family Abyssoanthidae, and we found these off Japan at 3,000 meters and 5,400 meters in the deep sea. Also reported from the Mediterranean at about 2,000 meters. Recently, we've also found them at 200 or 300 meters in Japan. But it looks like this is a common thing in the, not common, but occurs in the deep sea. But on top of this, we also found, so Abyssoanthus in 2007, we also found Microzoanthus, which is very small. This polyp that you're looking at, the diameter of the polyp is approximately 1 or 2 millimeters. And they're planktonovorous. They don't have any zooxanthellae. And then we also found even smaller Nanozoanthus with polyps with diameters of less than 1 millimeter that live in the sand on coral reefs and have zooxanthellae, Symbiodinaceae. And so finding these zoantharians was one thing, but then when we looked at the DNA sequences, what we found was, and so you can see this tree here, the blue is previously known Macronemina, the red is previously known Brachionemina, and these purple branches are all these new groups that we found. And so this really has sort of expanded our idea of all the different types of zoantharians out there. And so finding these 3 groups within 6 or 7 years was actually quite surprising, which really shows you how little people had actually gone into the field looking for this. Part of this is because scuba is relatively recent and access to the deep sea has been limited. But it suggests there's a lot more information out there and that our knowledge is incomplete. Then Swain and colleagues came along and did some excellent papers, a series of excellent papers using 16S, ITS, CO1 combined with morphology, detailed morphology. And so they suggested a deep sea origin of zoantharians and host associations. They suggested that host switching events between different host substrates is important in the evolution of zoantharians. And they also really re-examined the sphincter muscle and classified the sphincter muscle of these zoantharians, of their polyps, into 8 or 9 different types of sphincter muscles and overlaid this on top of their phylogeny. So these were really nice papers sort of combining the former morphology with what we knew about the molecular taxonomy or the molecular phylogeny. And then this year, we've had 2 big papers come out. The first was by Polaceno et al. And so this is a mitochondrial genome paper. So this is the entire mitogenome of 9 of the 10 families of zoantharians. We could not get Abyssoanthidae. And we found basically these results, again, show the problems with the suborder. You can see here, you have Hydrozoanthus grouping This is Hydrozoanthus here, grouping with this group of Brachynemina, but also sort of reaffirming the phylogenies that we've known until now. After this, the same year, or maybe last month, Quattrini et al., as part of their massive ultra-conserved elements paper, sampling pretty much all of Anthozoa, including octocorals and cereanthids and all the hexacorals. And we included 9 of the 10 Zoantharian families in here. There's a couple important results that came from this. The first is that in the hexacorals, which are from up here at the robust Zoantharia, Zoantharians appear to be the most ancestral of the hexacorals. Older than them appear to be the cereanthids. So Sergio's probably very happy. And as well, the octocorals appear to have a very different origin. But among the hexacorals, Zoantharians look to be ancestral. So this is one thing that's important. The other thing that was quite important for us was that we noticed that the mitochondrial genome, and this is a nuclear-based 1700 loci genome. It's very hard to see, but the phylogenies of Zoantharia do not agree. So there are some issues that we need to solve. This is also, again, very, very hard to see. The Zoantharians are over here, but what we found is that these newly discovered, the microzoanthids and the nanozoanthids, are based on this analysis, which is the most robust, ancestral, whereas in the mitochondrial, it's the deep sea ones. So it's clear that there are still some deep questions remaining in Zoantharia. The suborders still need to be reorganized, and there are some questions about what groups with what or what branched first. And so these are things that we need to work on going forward. So that's sort of an overview of the phylogeny. However, the result of this is, aside from the questions that remain, and again, these trees are just representations. Please do not get angry at the shape of these trees. This last tree here is just a representation. But until about 2007, in Zoantharia, we had 4 families and 10 genera. And we've been using museum field specimens combined with DNA sequencing, or now next generation sequencing. Currently, there are 10 families and 30 genera. And yes, some of this might be considered splitting of groups that were formerly large and sort of amorphous. But I would argue that this has really increased our understanding of the diversity of Zoantharians and how many different types of associations they have. They're a very diverse group with very deep branches, very long branches. And so some of these groups have been on their own evolutionary path for a long time. And so I think this makes this group, at least to me, something that's really, really interesting, not only at the deep sort of ancient levels, but also at the species level, because we see switching of hosts and lifestyles, even at the species level, which I'm going to talk about today. So our current taxonomic framework, we, as much as possible, in situ images are very good. Zoantharians, maybe some of you have information, but they're very stubborn, would be the best word. If you give them some type of relaxant or like magnesium chloride or something, often the polyps do not open. So in situ images, especially of healthy colonies, are very important. Host information, what animal are they attached to, if they're attached to an animal, and if you can get identification, very important. Morphological data set, especially the sphincter muscle, is good to see despite the problems with sand. We try to get sequences now of 6 DNA markers, 12S, 16S, CO1 from the mitochondrial, and then also 3 nuclear markers. But now we're also getting a lot more genomes and transcriptomes, SNPs, et cetera, to try to get more next generation sequencing data available. So that's sort of an overview of the taxonomy of the group. And what I'd like to do with the second half of my talk is sort of look at Zoantharian diversity research and taxonomy at 3 different levels. And so the first level I'm going to talk about is kind of at the, I guess you could say, the suborder or the genus level. And then from there, we're going to go down to species level. And then if we have time, inside the species. So this is work that I've been doing with Hiroki Kisei, who is a Ph.D. student in my lab, who will graduate this year. And he's been working on one of the suborders, the Macronemina. And so you can see in the background, there's a Epizoanthus iloricatus colony, and it's growing on top of a unicid worm, which is an annelid worm on the outside of the tube of this worm. And so these Macroneminas have associations with a lot of different animals. So again, yeah, here's your suborder. Zoantharia, currently 2 suborders, Brachynemina, which are the ones you commonly see on coral reefs, and then Macronemina, which associate with a lot of different animals. So today for this portion, we're gonna talk on the Macronemina. So Macronemina are found in most marine environments. There's 2 different types of lifestyles we can consider. So you have, this is an epizoanthus of some kind with a crab. This would be a pterozoanthidae of some kind with a sponge. This is another epizoanthidae with a annelid worm. But you also have some that are associated with, that just live on the substrate. So Hiroki's been calling these free living. To me, it sounds like they're out there, you know, no corona restrictions, having fun, but they're non-associated would be the best way to explain it. And there's 153 species at least of Macronemina as we currently know it. So we looked at the evolutionary transition of lifestyle between associations and non-associations and between hosts. There was only one study on this. It was done by Swain, nice paper in 2010. But since then, we've discovered a lot of new taxa, a lot of new groups. And so we have a larger sampling set, and we have a lot more information on the associations. So we decided to take another look at the associations and see what we could find. So Hiroki has been looking at species. He's been describing species. He's been also trying to construct a robust phylogeny using as much data and as many specimens as he can, and then looking at the evolutionary transitions. So we've been collecting specimens from all over the world for the past 15 or 20 years. But also this year we had planned to go to many museums to look at type specimens. Obviously that's not working. But we looked at external morphology, tentacle number, size, etc. Internal morphology, numbers of mesenteries, the cnidae, things like that, the sphincter muscle shape. And then for molecular phylogenetic analyses, we did the 6 markers and we did ancestral state reconstruction. As the results, so Hiroki, during his PhD, he has described currently 12 species, I think, including 1 genus. This one in the middle here, Enigmanthus, was a new genus described last year from Japan. But we've also found species in the Caribbean and around Australia too. So it's clear that there's a lot of species out there that people haven't noticed or documented. In other words, what we can take from this is the species diversity of Macronemina, of the suborder Macronemina, is probably much higher than the literature suggests. And this is something that other people have also found in the deep sea. Basically, if you find a zoantharian from the deep sea, there's a, well, I don't want to say a great chance, but there's a fair chance that this is something that is undescribed. So these deep sea specimens really harbor a lot of diversity. When we look at his tree, and I'm just going to show you parts of his tree today for the sake of discussion. He focused on Macronemina, and one of the families in there is the Epizoanthidae. You would know these as the ones that associate either on annelid worms. You see them in associations with hermit crabs, with mollusks. You also find these on the stems of glass sponges. And so when he reconstructed a phylogeny with all of his different Epizoanthidae specimens, plus this weird group here called Isozoanthus that was described in 1923 off South Africa. It's huge. It lives in the sand like a bowling pin. And this is the best figure we have of it from this 1923 paper. So this is sort of a mystery, but we do have some DNA sequences from more recently collected specimens. So what he found was that there were 3 different clades of epizoanthids, as well as Isozoanthus being very unique in its phylogenetic position, in its morphology, and its ecology. We knew about the morphology and ecology. Basically, this Isozoanthus group, we believe, should be its own family. Aside from having this very weird lifestyle of free living, it's a brooder. It actually is very unique among zoantharians that most are broadcast spawners. This species is a brooder, so that's something that's really unusual. And then also inside the Epizoanthidae, based on molecular distance, but not only molecular distance, but also association, it's clear that there are 3 clades within Epizoanthus. We think there's 2 undescribed genera inside this group. We have one group that associates with sponges and with annelids. You have one group, this is the one that we're going to call Epizoanthus, that associates with mollusks and crabs. and then another group that only associates with glass sponges. So within these groups, you do see some sort of specificity about what host animals they associate with. And the same, when we looked at what we thought were Epizoanthidae, we actually found that some glass sponge associated species are not Epizoanthidae, but are in a totally different family, the Parazoanthidae. And so looking at their histology, they have sphincter muscles like the Parazoanthidae. And when you run the phylogeny, they do not match with anything that we've known inside the Parazoanthidae. The associations of glass sponges were only known from Epizoanthidae. So again, there's more diversity. In other words, these associations are specific to clades, but inside the entire suborder tree, they might appear at multiple spots. So it's much more complex than we had originally thought. But biodiversity usually is this way. The more you dig, the more you find. So we've got a lot of work ahead of us still. When we did ancestral reconstruction, what we found was it was suggesting that if you look over on the extreme left, you have this free living group here, especially the Isozoanthus. And so this is suggesting that the free living group was the most ancestral. This is very different than previous ones that has suggested the epizoanthids, the hermit crabs and the annelids were ancestral. And it shows that taxonomic sampling, wide sampling is very important. However, I would take this with a grain of salt because there might be something else out here that's that we don't have information on that might change the most ancestral state again. But the other thing I think that you can see from this ancestral state reconstruction tree that's very clear, you've got associations with 7 or 8 different phyla, and most clades have pretty clear patterns, but you can see some species switch. For example, if you look at this group here, you have groups associated with annelids, you have groups associated with hermit crabs, and you have a few species associated with mollusks. Over here, you have one free species that just appeared sort of randomly. And so you have, it appears, you know, the ability to associate with different phyla, and these branches are deep, but occasionally you have very shallow recent switches to different hosts. And so we speculate perhaps the free living mode was ancestral, symbiotic mode is more evolved. But the evolution of lifestyle can be explained by the results of ecological adaptation. So most zoantharians, especially deep sea zoantharians, are filter feeders. Only shallow water species, Brachynomena, different suborder, have zooxanthellae. So we speculate that this association with other hosts allows them to obtain, you know, better, safer, more stable nutrients. And so this is something I think that we need to look at going forward and consider a little bit more. So in conclusion, diversity of the suborder is much higher than we thought. The ancestor of the macronemic zoantharians might need to be reconsidered. And we also, this is sort of supporting what Swain also had found earlier, interphylum host switching events look to be key for diversification of this group of animals. So this is something that's ongoing. There'll be more papers coming out. But again, if you have any unusual or strange macronemenic specimens or information, I'd love to talk to you. It'd be really interesting to talk to you. So we've talked a bit about genera and families, but now let's look at a bit closer, species. And so we're gonna switch suborders. We're gonna switch to the Brachynomena. These are the zooxanthellate, the coral reef, shallow water species. And we're specifically going to look at Palithoa, which is very common in the Atlantic. It's common in the Indo-Pacific, probably the most common one that you will see, group you will see on coral reefs. And so this is work that's been done by Mizuyama Masaru. He just recently graduated from our lab, is doing a postdoc up in Tokyo. And this is a lot of this part of his talk is based on his recent research in his Ph.D. Sorry, close the window, jets outside. Okay, so he's been working on coral reefs, and so Palithoa are found in the intertidal zone. We all know that the intertidal zone is very stressful for animals. And also there's very high biodiversity in the intertidal zone. And so a lot of recent papers have looked at sympatric speciation, or speciation of species within the same location or environment, in the lack of geographic or large-scale barriers. And coral reefs really seem perhaps to be major generators of marine biodiversity. When you look at Cnidaria, there's actually, the species question is something that really troubles all of us. There's been a lot of work on Acropora with cross-fertilization, but a lot of our animals, as you know, are non-model organisms. A lot of these animals, including Palithoa, including a lot of these scleractinian corals, spawn once a year. And in Okinawa, if there's a typhoon or bad weather, you miss the spawning, you wait another year. So it's hard work, a lot of field work, but integrative approaches using morphology, ecology, reproduction, and molecular data sets have been successful in sort of teasing apart species relations. And so this is what Masaru aimed to do with some of the Palithoa. So Palithoa, I'm sure you're familiar with Palithoa. You've probably seen these species. Palithoa keterborium is very common in the Atlantic. They are very, very common brachypneumatic zoantharians. They have an amazing amount of sand uptake. There are 272 names in the literature, although how many are valid is an open question. In Okinawa, we commonly see 7 different species groups. And so what Masaru decided to do was we focused on 2 species that we know have a very close phylogenetic relationship. We focused on Palithoa tuberculosa, which is sister to Palithoa keterborium, looks very similar. And we also looked at another one, Palithoa mutuki, which has very different sort of freestanding polyps connected by a stolon. And we know they're closely related because in 2007, we published a paper showing they were closely related and also showing the presence of a third species or species group or something. This Palithoa species Yoron, it was intermediate in morphology between mutuki and tuberculosa, intermediate in phylogenetic signal. So we speculated maybe this had some sort of hybrid origin. I can tell you details if you have questions about this after, but it was clear that this was a messy species group and that it needed more information, and DNA alone was not solving the question. So what we did was we investigated the ecology of species Yoron and tuberculosa and mutuki, and you could see that they all lived sympatrically on the same reef, but in slightly different microenvironments. The morphology was also clearly different. So we really had a lot of questions about these 2 or 3 species. And so what we decided to do was we decided to take a look at their spawning timing. Is there a chance that there could be some sort of cross-fertilization? We decided to look at species boundaries using molecular phylogenetics. And then we also decided, if we decided they were different species, how could we explain how these species had recently evolved? So morphology, spawning period, and molecular phylogenetics were the things that we looked at. So what we did was, this is a map. Sorry, there's no world map. This is the bottom big island is Okinawa. That's where I am right now, talking to you from Okinawa. And Okinawa and the Ryukyu Islands is actually a chain from mainland Japan all the way down to Taiwan. So there's a lot of different environments, but this time we focused on one island here, Tokunoshima Island. Tokunoshima Island is really unique because the intertidal zone is quite pristine, and you can find different Palithoa living together in the same intertidal zone. And while we were doing our initial research, we actually found another species or morphotype, which we called Palithoa aphmutuki, because it looked like Palithoa mutuki, but it was different in tentacle number. It was different in the arrangement of, it didn't have any capitulary. You can look at the photograph and see there's no clear ridges here, like as in mutuki. So yes, it was clear that we were dealing with some kind of species complex. So what we did is we looked at numbers of tentacles. We looked at the cnidae under the microscope, different types of cnidae, and measured the different cnidae, the lengths, the widths, the abundance. And we looked at the mesenteries under microscope to see when their spawning timing was, and did a lot of field work. And as well, we did DNA extraction and alignment, and we looked at 4 markers, CO1, 16S, which are mitochondrial, ITS, and ALG11, which are nuclear, and made an alignment and did some phylogenetic tree reconstructions. So what we found was actually pretty interesting. Morphologically speaking, you can clearly delineate 4 groups. The numbers of tentacles are clearly different, and the numbers of mesenteries are clearly different for all of the groups. If you looked at the Cnidae, however, there were very subtle differences. For example, in the column and in the tentacles column and pharynx of Mutuki and Aphmutuki, we could find small holotrichs, which we could not find in Tuberculosa or Euron. But other than that, not many differences. And this is to be expected. Zoantharians have 5 or 6, depending on who you talk to, types of nematocysts. And there's not like anemones, there's not a large repertoire within them. When we looked at spawning timing, again, what we found was reproductive isolation. Paleotuberculosa looked to spawn around the end of July and August and early August. Euron spawned in early to middle November. Aphmutuki in the middle to late June. And then for Mutuki, the species, not Aphmutuki, we actually didn't find much information about spawning at all. Whether we just had bad luck and there was no spawning that year, we don't know. We actually looked at 2 different years. When we looked at the sequences, what we found was based on the sequences we had, the mitochondrial DNA gave us either 1 or 2 groups on CO1, 16S again, 2 groups, ITS, 2 messier groups, and then ALG11, everything was completely mixed mashed together. So each marker alone really didn't give us that much information, but when we concatenated it and did a species tree estimation, what we found was we had 2 different models that were equally well supported, 4 species or 2 species. So in other words, DNA alone was not giving us the answer we needed. It's like, oh, maybe 2, maybe 4, which we kind of already knew. So when we consider this, okay, morphology, clearly different. The problem is when you go to the literature, for example, looking at numbers of tentacles, we found very clear numbers for Paleotuberculosa, 31, 32, 33 tentacles. In the literature, the numbers are 30 to 52. Mutuki in the literature, 42 to 144 tentacles. So in other words, we have clear delineations of our specimens in Japan, but when we consider to the original literature, they're all within the range of what these species have been described at, which indicates that previous literature might have lumped things together, or that these are just morphotypes. In other words, it's a bit of a mess. As well, there's been research that has shown that Paleotuberculosa is phenotypically plastic. So we found some Cnidae results as well that showed very fine scale, but nothing clearly diagnostic. Spawning period, on the other time, this was a lot of work, a lot of hard work, but we found for 3 of the 4 groups very clear separations of their spawning timing. This sort of compared well with previous research, but a little bit different. But what we think is it looks like there's some kind of reproductive isolation, probably temporal, going on. So we think that currently natural hybridization between these closely related species is probably not occurring. And finally, when we look at the phylogeny, 4 species and 2 species are equally well supported. At least there are 2 groups here, maybe 4, but within these 2 groups, we don't think there's any genetic exchange. So the results imply within the groups, it looks like maybe those groups are not completely separated. There might be incomplete lineage sorting or introgression of some kind. So in summary, what we can say is looking at these species groups, do they have a chance for in situ interspecific cross-fertilization? We think probably, at least for Japan, no. Where are the species boundaries of this species group? If we look at the morphology and the spawning period, which I'm going to go with because I think this is a much clearer indication, we think there's probably 4 species here. So how do they speciate sympatrically? They're found on the same reef, in the same location. So we sort of suggest that the niches that they actually live in are slightly different on their reef. The abundances are different in different locations. As well, data I did not show you today also shows that they have, the 2 groups have distinct, slightly different Symbiodinaceae. So we think that if you actually look at a reef, and this is something that I took from a Japanese textbook, but I think it shows it well. This is a typical fringing reef in Okinawa. There's a lot of Japanese here, but what it's actually showing is, and this is something we know from working on coral reefs, is even within a reef that's 50 or 100 or 200 meters wide, there's a lot of very different environments with different lighting, different currents, probably different amounts of nutrition arrive. And so you see very clear differences in the species that are present there. And so I think that the complexity of these coral reefs might be acting as a recent generator of species diversity in Palithoa. Just for one last thing, just to quickly show you, we actually have done some next generation sequencing as well. And the results, in short, are a mess. Next generation, this is actually SNPs. So I think this is around 245 biallelic loci generated from EasyRad method. And what this is showing, this top group here, all the way from the yellow Caraborium, tuberculosa, Euron, tuberculosa, Mutuki, grandiflora, these are all Palithoa. And the thing that I find interesting is you have an Atlantic species, Caraborium, placed inside this very large Indo-Pacific species group. And this is different than what you see in Scleractinia and a lot of other zooxanthellate anthozoan groups. And this is implying that we really don't understand recent species evolution in zoantharians clearly. It's also open for debate as to whether this is one species or several species. I've been working a lot with Rob Tunin, who's done this for a lot of different animals, and he's never seen patterns like this with such a mess. So in other words, the good news is, or the bad news is Zoantharia is still a mess. I'm going to skip a few slides because I wanted to talk for 45 minutes. I have a chapter on nutrient acquisition and stable isotope analyses, but I'm going to skip over that today in the interest of time. But if you have questions about stable isotope analyses or nutrient acquisition, please ask during the questions and answers. But finally, I'd like to talk a bit about conclusions and the future of zoantharian research. So to start with, okay, what do we know? So we know this. We know that molecular and ecological and the morphological framework works well. We've got a pretty good understanding of zoantharian systematics. It's pretty easy to distinguish genera and families, straightforward. We also know that the macronomina, the groups that are often associated with other organisms, there's a lot of undiscovered diversity, especially in the deep sea or in unexamined locations, which means that fieldwork's important. But we've also made a lot of large discoveries in the last 10 years, which means that we actually don't know much. In other words, there's still a lot of diversity out there remaining to be examined. There's also a lot of remaining questions. For me, the big questions are at either end of the taxonomy. So at the species level, there's a lot of questions, obviously, but there's also a lot of questions at the high end, at the suborder or higher. The mitochondrial and the nuclear data sets do not agree. And I think, as Sergio Stamper could tell you, mitochondrial DNA evolution is weird. And so something has happened in the evolution of zoantharians, at least in their mitochondrial data set, that did not happen in their nuclear data set. And so that's something we need to consider. As well, subordinal taxonomic reorganization is needed. There are still only 2 suborders. We know there are 4 or 5, but the data sets are not agreeing. So we're trying to work with that, plus the morphology, to make new or different suborders. And brachionomina, the ones you see on the coral reef in terms of species, I wrote there, it's a hot mess. It's a hot mess. There's a lot of literature. A lot of descriptions are vague, and the DNA is actually not really clarifying a lot of the questions we have. Some of them it is, but some of them it's not. So we're still working on a lot of this. So the genetic and even the genomic data, often they're not solving these species questions. So reproductive information, morphology, combined approaches, I think, are needed to really sort of get at the next level of zoantharian systematics and evolution. So with that, I think I'm done. I'd like to say arigatou gozaimasu to all of you for staying awake. and to all the LANIDA people, and to Sergio and the organizing committee for inviting me. I'd like to thank all the collaborators, JSPS, which is our national funding institute here in Japan. We've received a lot of funding from a lot of different agencies to do work overseas. And if you're interested in collaboration, please email me or Twitter me or whatever you like. And I'd also like to just give a quick shout out to these 4 young researchers, Angelo Poliseno, Masaru Mizuyama, Maria Santos, and Hiroki Kise. They're all excellent researchers who I think are going to do great things for Zoantharia and Cnidaria in the future. And so with that, I would like to say thank you very much. And I'm going to stop my slide share there. And maybe, I hope I didn't speak too quickly. There are some questions. Thank you very much.
Okay. Do you see me? Yeah,
Yeah, I can see you. I can see you. No worries, I can see you.
Well, thank you so much. The talk was awesome. It was incredible. It was very interesting. Nobody is going to sleep, so that is really
I hope not. I drank my coffee to stay awake.
No, it's early for us, for me, for example. And you generate a lot of questions, so I'm going to start to asking you. But first, I really appreciate your participation here in this initiative formulated by the network in commemoration of the anniversary of it. So thank you so much for participating. And I'm going to read you the greetings and the questions that everyone was asking in the Facebook Live. Well, we have different ratings from different parts of the world, like Sao Paulo, Argentina, Mexico, and there is people that I don't know where they're from, but they speak in English and in Spanish. So
no worries.
different parts of the world. Well, the first question that I have is where we can find your job about DNA taxonomic.
Where, sorry?
Where we can find the job, the paper that you write.
Oh, okay. Yeah, so that's the good thing and the bad thing. I think there's been Senegar, Swain, myself, also, I think there's been a lot of papers that have been published on molecular methodology. Basically, most of the papers use very similar methodology. We all use now pretty much a standard set of markers. If you want the most robust information with the 6 markers, Kise's recent papers, if you search on Google Scholar Kise and Zoantharia, maybe his 2017, 18, 19 papers should be pretty robust, but we don't have how can I say, we haven't made one large paper like an overview on this. It's always evolving a little bit. But I'd say look at the Kise papers, and if you have any questions, email me, Spanish, English, Japanese, anything okay.
Japanese is a little bit complicated, but English and Spanish goes better. Well, Juan Tobar asked, why are nidocysts or nidoma not so widely used in description of zoanthids?
Okay, so why are they not widely used in the taxonomy, you meant?
Yes, I suppose.
Yeah, okay, and they're in the systematic. So there is a group of researchers that is trying or is using them. Ocaña and Brito have published a couple of papers using nematocysts. However, compared to, for example, anemones, or maybe even cereanthids, the diversity of types of nematocysts is not very high. And the nematocysts that you see are not very big, which indicates they're not really, they're filter feeders, but they seem to be targeting, I think, different groups of organisms compared to maybe some of these more aggressive animals. So, and then the other thing that we found, we have unpublished data, I think probably Fabian Acuna could tell you more maybe, but we also see that there is a little bit of variation in the same species in different locations. So we have nematocyst information in all of our descriptions because you need to describe the animal, but I think we don't want to use it to say this is different than this species, because we're really not sure if it's diagnostic or not. So if that makes sense. So it's good information to have, and you need to include it, just like the color, the size, the location, whatever. But to use that alone to say this shows that this is different than this, to me is a little bit risky, if that makes sense.
Yeah, maybe it's a little bit complicated, and there are another characteristics that are better to.
Yeah, that's the problem that's been facing Zoantharians throughout their history is, as a group, they look very similar to each other. The matocysts look similar. The sphincter muscle looks to be different, but there's the sand issue of cutting. So, but there's no clear. We don't really have a beautiful smoking gun that if you look at this, you know the species. We have to combine a lot of different data sets, and then we have to think and maybe drink a survey. So I don't know, but it's a lot of it is it. But I think this is common though with marine species. In the end, species is a biological unit, but we're the people that are judging species or making decisions. And with a lot of these marine groups, it's really hard initially to tell what is important and not important. And so Swain has done some very nice analyses of the different characters and what could be important. And even then, you look at the former researchers, you know, like Haddon, Shackleton, even Ellis, 1768. They actually did a pretty good job with no DNA of figuring out the groups. So we're all in the same boat. Species are tough.
Yeah, but it's complicated sometimes to identify, and sometimes one character works very good for one and for other.
Yes. Yeah, totally. We have a couple species. Oh, this color is this species. But if you use color for all species, it's a disaster, right? So yeah, there's no perfect answer.
Okay, thank you.
No, thank you.
We have another question that said, are you used to work to generate works about the interactions with other animals? And if it exists something similar, another study similar about the phenomenon of coevolution, that if you
coevolution. Okay, yeah. So this is something that we've sort of dabbled with. I think evolutionarily speaking, it's clear that these associations between different phyla are really important. And when you look at the phylogenetic tree, the branches are very long. So this is suggesting a long history of association. And these animals that they're associating with include echinoderms and annelids and mollusks and crustaceans and hydroids. And these are all animals that have a lot of different chemicals. So this is really implying a long history of coevolution. But the crazy thing is you see sometimes there are some species that can associate with 5 different phyla, a few species. And then you see some species that switch. So all of this group associates with this phyla, but this one species associates with this phyla. To me, that suggests that a lot of these zoantharians, they have like a toolkit of all the different phyla they can associate with. And these switches can go off and on like in evolutionary time very quickly. So to me, you know, I'm not a genomics expert. I'm not a functional genomics expert, but trying to figure out coevolution and how these animals can get around the chemical signals that prevent other animals from settling on these host species, etc. I mean, yeah, there's basically, I have to live to be about 500 years old, I think. There's so much work out there. But this is something I'm really interested in.
Yeah, it's coevolution and all of that is really, really interesting. And in these groups, I think that it's more interesting because the animals adapt very, very fast, as you was saying.
Yeah.
So it's going to change maybe in a minute. So it's more complicated. Okay. Guy Alvarez is asking if you can talk a little bit about the biogeography of zoanthids, if there is a distribution pattern, or if it is data that is still missing.
Okay, so yes and no. You are correct in that there's a lot of locations where we do not have information. Particularly the Coral Triangle for us is very much a black hole. We have some information from Indonesia, we have some information from the Philippines, but we've never been able to really do a targeted search there. And the countries have very restrictive policies in terms of biodiversity research, which we're all familiar with. So for shallow coral reef stuff, we suppose that the central Indo-Pacific is the center of biodiversity, similar to corals and many other coral reef species, but we don't know for sure. The current data is southern Japan, but I'm sure that's biased because we do a lot of work in southern Japan. So for coral reefs, we sort of believe it follows the fishes and corals pattern, but that last slide I just showed you suggests that the Atlantic and Pacific and the whole closing of the Isthmus of Panama, something different has happened with some of the, with Palithoa and probably Zoanthus. And because they're zooxanthellates, we don't think they've gone around South Africa or around Southern America, but there's a few patterns we don't understand. So yeah, biogeography is still in its nascent stages. I can tell you that Zoanthus and Palithoa, and we've published papers on this, there are sibling species between the Atlantic and Pacific. This looks to be very clear. And there's a few species only in the Atlantic and a few species only in the Pacific. So some of these patterns fit. But then for the other group, the Macronemina, the deep sea ones, no idea. Basically, yeah. I just did a review paper. We were looking at north of Japan and then the Kamchatka Peninsula, and we were looking at the reports on Cnidaria from the deep sea. So this is an area the size of France, right? And
also, it's more complicated to take the
and
animals to know
hard
where
access.
they are.
Yeah, it's crazy. And so we found, I think, 180 Cnidarian records from the entire region through all of history. And it's you know, it's, so there's such one cruise, one boat trip with a net to 1,000 meters pulling up stuff could double your information.
Yeah,
right?
Exactly.
So yeah.
Well, thank you. I have another question of Naleli Ruiz.
Yeah.
You know her?
Oh, yeah. I think so, yes.
Each character within the character complex can be evolutionarily modified independently of zoanthids, or
so you mean can each character be modified? So within a species complex, can characters be modified independently? We don't really know. I think this is related to function. We've done, sorry, I keep opening and closing my window because we're by a US military base and they keep flying planes by. So
nobody knows there is a window there.
Yeah. Oh, see, looks like the ocean, right? Yeah. So I think in terms of, the first thing is we need to know the function of each character. For example, tentacle length. Tentacles grow longer in areas with low current, but they also grow longer in species that live in caves when they lose their zooxanthellae, which implies that the longer tentacles help them feed more. So some of these zooxanthellate species seem to have shorter tentacles. So low light or low current, they seem to get bigger. We also know that in low light, colonies spread out more. And the oral discs seem to get bigger, maybe to attract more light. So some of the groups we've been working on for functional morphology have been Zoanthus and Palithoa mainly, because they're easier to keep in tanks and easier to do experiments, and they're easier to sample. So whether they're independent, I don't think currently we can 100% answer that, but I don't think they're independent. I think a lot of these are related to feeding. A few of these are probably related to maybe defense. A few of these are related to reproduction, but I think they're linked. But within this linkage, I think the thing that has made a lot of these coral reef species so successful is they're so plastic. You can take a colony and put it in a tank and turn the lights off for 6 months. And as long as there's fresh water and some plankton in there, they survive, they're fine. And the colony will have gone from thin, the polyps will be long and skinny looking for light. So these things not all, but many of these species seem to be very generalist, that are very able to change their morphology in reaction to their local environment. And we see that in the chapter I didn't talk about with the stable isotopes. They can change their modes of feeding depending on depth.
Oh my God.
Yeah,
That
yeah.
is really quickly, or more or less quickly, that they
Yeah.
adapt for these.
They're very good at local adaptation. I would, not all species, but a lot of species, yes.
Okay, thank you.
No worries, thank you.
We have another question about characters that says,
Yeah.
so the sympactor muscle would be the best morphological character used to Zoantharia now?
So yes and no. The sphincter muscle, I would say, is the best for, not for species, but it can place them in groups very well. So like we know this genus has this type of sphincter muscle. We know this genus has this type of sphincter muscle. So I think in terms of placing, some of these groups are very hard to tell apart without sphincter muscle information. So in that sense, yes, to genus, but then for species, sometimes no. And then the other thing is, due to incrustation of sand and detritus, it's a very important morphological character, but it's often very hard to get information on it because you have to do the decalcification or the desilification properly. And if you don't, you just destroy your
everything.
Blood. Yeah. So it's very important, but it's often hard to get. And also for us, like for example, nanozoanthus, less than 1 millimeter in diameter, orienting the polyp in your wax to get the proper cut is, it's just like, it's like rolling 2 dice and hoping for 2 6s. So please, you know, it's so random.
No, for me, it's impossible to that.
Yeah, no, I mean, it's possible. It just takes a lot of work and a lot of time. I think Swain really has shown the importance of sphincter muscles. Sinninger showed the importance of host associations. You combine that with the molecular data, and it's evolution. You know, it's a mess, right? But it's fun.
Well, thank you. We have another question of Tiago Benicius. He said, hello, Raeimer. Thank you for sharing your knowledge with us, but I still have a doubt. In addition to symbiosis, what other ecological factors can integrate data that assist in the taxonomic of macronemic zoanthids?
Ooh, so
very deep, the question.
Yeah, deep. I need more coffee. Let me think. Okay, so there are other things, there are other data sets that we haven't looked at that we could probably look at. I think looking at I think there have been a few papers that have looked at chemoecology to some degree, or chemotype. You could look at the shared marine natural products between different hosts and the zoantharians. That might give you some hints or about associations. You could also maybe look at, we look a lot at the symbionts. We look a lot at, for species that have zooxanthellae, that have Symbiodinaceae, we look at the Symbiodinaceae. But we haven't really looked much at the microbiome. We haven't looked at all the bacteria or the viruses. So there might be something there. We've also done a lot of CT scanning recently. We've been using micro CTs to look at how the host, whether the host deforms its shape to accommodate the zoantharian. But surprisingly, usually it looks like there's not much deformation. It looks like they're pretty happy with each other. Occasionally you see it on the octocoral, the precious coral associated groups. You see scarring and pits, which might indicate, a parasitic relationship. But generally it doesn't seem to be. There's no, not yet. But I'd be happy to hear your ideas. Like if you have some. I think I answered his question.
Yeah. For me, yes. I don't know from him, but for me, yes.
Okay.
Luke Jimenez said, first, thank you so much, James, for the interesting and the clear talk. Do you have any information about records of non-native zoantharian species?
Okay. So invasive species, in other words. So currently, yes and no. Yes. Let's say yes. Okay. So generally speaking, there hasn't been much information. Like I know octocorals are a big problem in the Atlantic right now. But I also know that Carajoa in the Pacific, which was thought to be an Atlantic species, actually is a Pacific species.
Yeah.
But these are octocorals. For zoantharians, I can give you two examples of well, one of invasive and one of biofouling. So we know that some zoantharians like to live on artificial substrates to some degree. And so we found a lot on pipes in Palau. So often you see anemones in your tanks and on your pipes, but in Palau at this one sort of, they were pumping in seawater and all of their pipes were coated in microzoanthus, which was kind of cool for us. But that's not invasive, that's just more biofouling, like nuisance.
Yeah.
The only invasive species information that I have involves Honolulu. In the harbor there is a species, an undescribed Hydrozoanthidae species that's in the aquarium trade called yellow polyps. We know it's an undescribed species. We've never been able to describe it because we can't find it anywhere except Indonesia, and Indonesia is quite difficult to do work. But it looks like there are yellow polyps all over Pearl Harbor. And so people speculate, these things are traded in the aquarium trade. So people speculate that maybe it's an invasive species, but the problem is it's unlikely, but they might have always been there, you know, or they might have been there and not common and now they're common for some reason. I don't know, but to me, Hawaii and Indonesia seems a very disjunct distribution. So I'd say probably it's invasive of some kind, but no information, no one's done any research on it. I've just sort of basically heard from fellow divers, basically.
I think that it's very difficult to know that also because maybe someone discovered there is a new species or something in other part of the world, and maybe that is always there. So that is very complicated because maybe you think that there is an invasion of someone of some species.
No, it's tough, right? Because like, for example, Terpios, which is a sponge that kills corals, we found it in the Great Barrier Reef for the first time. Then we found it in the Maldives for the first time, but it might have always been there. And now it's just increasing due to climate change. So there's a lot of, I think, it's really hard to say this is invasive. I think the octocorals, the Atlantic Pacific is a very clear case. But within the same ocean basin, you know, there's so little data out there, you know, we need so much more information. It's tough. Yeah. But fun.
We have another topic to know now.
Yeah, more research.
Yeah, we need some more people to work.
No, we're never going to run out of questions. We're never going to run out of science. That's the good news.
I hope that.
Yeah, no worries.
Well, Iris Sambayo said, outstanding talk, one of the great opportunity of Zoom talks, thanks Red Lanida and Dr. James Raeimer. In such a mess, taxonomy, how or when do you make a decision of species imnotaus? Okay, so good question. A question that we often think about. And I think there's two ways of thinking about things. So I do my research in Japan, and I know a lot of Japanese taxonomists. And in Japan, making a mistake is very embarrassing. And so a lot of my colleagues are very, very, very cautious about taxonomy. I'm not quite so cautious because maybe I'm more cautious with age than I used to be, but my thinking is taxonomy is a tool. And it's a tool to put a name on something, of what we think is a biological unit of some kind. And so if we know that this is a different species, or we think it's a different species, and we don't put a name on it, I think it makes it harder to track things down in the future. In the end, we might be wrong. Maybe it's already been described, or maybe it's actually a different morphotype of something else. So you need to do all that you can to show that this is new or different. But taxonomy is full of mistakes in the past, and no one has access to all the data. And I think if you're willing to make some mistakes,
you don't set out to make mistakes. I think you want to do your best, but taxonomy should be a living thing, and it should, and it's imperfect. Science is imperfect. We're always improving. And I think we postulate a theory, we put it out there, and if we're proven wrong, c'est la vie, you've helped move science forward. And so I'm kind of the living, I believe in living taxonomy. It's something that's changing, that's evolving. And I think some people have a different way of thinking about that. That said, of course, you want to do good descriptions, you want to have type localities, type specimens, all of that. You need to follow the rules, obviously. But science is not going to move forward if we just sit on data because we're worried about it. So if you're really uncertain, you don't do anything. But if you think, well, let's get some more data. Okay, let's get more data. Okay, well, I have some species or not species that I'm sitting on because I'm really not sure. But there's others that, we think they are good species. 20 years from now, who knows?
Yeah, it is about what we were talking about recently, about that. Yeah. Maybe someone described in one place, another in other, and the names change all the time. And so taxonomic is very changing science, I think.
Yeah,
it's and that's changing, and it's not wrong.
It's not static. No, and that's good. I mean, the more people that are working on things and the more people that are putting out ideas out there, in the end, things will slowly become organized, right? There might be, wow, but then after it makes you look and reconsider things. So I think the more taxonomy that's being done, the better. And I might not agree with all the taxonomy, and people might not agree with my taxonomy, but that's okay. That's science.
It's normal. We're humans.
Yeah. Exactly.
Well, I have another question of Roger Ayres. It said, why the hot mess in Brachynemia and the deep diversity of Macronemia, which is the most challenging suborder now?
Woo! Okay. Macronemia is challenging because we keep finding new things, and there's so many new things to be found out there. And hi, Roger. So that's cool. But that said, when we receive or when we get specimens of Macronemia, DNA combined with host information, we usually can figure out pretty rapidly what this is and where it belongs, whether it's new or not. Because historically, there's not a lot of literature on Macronemia. There's not so many comparisons to make. For Brachynemia, most of the species are shallow. There's, Paleozoa, 270 species in there. And so we have a lot of species, even in Japan, 3 or 4 of these species that I was talking about. We know the molecular, we know the morphology, but it fits with like 5 or 6 or 10 species in the literature, and the type localities are all over the place. So in order for us to really sort that out, we either have to look at the type specimens, and now we cannot because of Corona, but we couldn't before because many of them were lost in World War II in Germany. Or we need to go to the type localities again and collect neotypes or topotypes, which is also now in 2020 not so easy, and also costs a lot of money and time. So
and it's complicated for sometimes to find. Maybe was one chance to find it. So
but it needs to be done. So I'm hoping. I just put in an application to actually go to all of the type localities for all of the Paleozoa and all of the Zoanthus, or get people to collect and to do genomic analyses of neotypes of every single species out there. Probably impossible, but
it's a good dream.
Yeah. So in that sense, I'd say Brachynemia is a hot mess. We haven't really formally described species since 2006 because I realized it's a hot mess, and we know what species groups are out there, but we need more information to reconcile. So in that sense, what do? We're sitting on information, trying to figure out what to do. But for the Macronemia, green light, go.
Okay, well, and we have the last question. I said,
okay, sure.
Why did you start working with Zoantharians? What take you there?
Yeah, so this is actually a whole nother PowerPoint presentation. I was not originally interested in zoantharians. I was in Kagoshima, which is in southern Japan, but maybe subtropical temperate on the edge. So no coral reefs, but there are corals, zoanthalate corals there. And this was I was doing my master's. I think I started in like 1998, many, many years ago. And there was a big coral bleaching event. El Nino was happening. And so this was the first global sort of bleaching event. And so for my master's, I wanted to study coral bleaching in Kagoshima. But there were not many corals and you needed permits. There were many octocorals, but they had no Symbiodinaceae. So one of my colleagues, a guy named Ono Shunsuke, who I've written some papers with, he was like, hey, why don't you study zoantharians? He said this in Japanese, and I had no idea what Tsunaginchaku were. I was like, oh, sure. But that turned out to be zoanthus. And so I was doing my master's on zoanthus and on zooxanthellae condition in seasons in zoanthus. And during my midterm master's presentation, so after 1 year, you have to give a presentation, and senseis, other professors, you know, they give you comments and say you're bad, or, you know, whatever, or you're good, whatever. And one of them said, what species are you working with? And I had a guidebook, a Japanese guidebook, you know, like a diver's guidebook. And I was like, oh, it's this species. And they're like, you can't trust a guidebook. You know, how did you ID it? And I was like, the guidebook.
No.
So
trust in what I read.
Yeah. So I realized, they said, you know, you need to ID your animal better. And so I started to email, and I started to look around, and I contacted John Ryland, and I contacted like Yoshi Loya, and I think Burnett was still doing work at that point in time. And everyone said, oh, zoanthus. And so I was so surprised. I realized that no one in the world could identify these species. And all of a sudden, I had this huge, what species? I don't even know what species. So that it was necessity. And so that's what started me on zoantharians. And then from there,
way to start.
Panic, basically.
Apart in the moment that everyone is talking about what you're doing, is worse. You get more nervous.
No, it's, I mean, it was basically an open niche. And I didn't set out to be a zoantharian expert. I set out to understand coral bleaching. We still do bleaching and coral reef research in the lab, but yeah, it just sort of happened. No plans.
No problem. Well, thank you so much, James, really. Thank you for answering all the questions. And if you want to give us a message for everyone that is watching, we are very thankful.
Okay, cool. No, I'd be super happy to. So for everyone watching, I'm assuming or hoping that you're all big Cnidarian fans. And I'd like to sort of say two things. The first thing is research on Cnidarians. You know, many people might say, what is a Cnidarian, or what are you doing, or why do you like these animals? But I disagree. I think biodiversity is very important. I think biodiversity of Cnidarians is very important. And I think understanding Cnidarians is very important. And I think it's going to become more important in the future. So for all of you studying or researching or working on these groups of animals, I'd say keep going. Your work is important. People do notice your work. I think it's something that is going to become more important in the future. And so I'd like to say, you know, ganbatte kudasai, which is basically work hard, don't give up. And then the last thing I'd like to say is related to that. Many people might say, you can't make a career doing this, or you can't find a job doing this, or you won't be able to amount to anything doing this. Because everyone told me that too. Everyone's like, why are you studying this group of animals? But again, I disagree. And I think with teamwork, collaboration, and a bit of luck, everything should be fine. So my advice to you guys, to everyone out there, is love your Cnidaria, do your work, and enjoy it. And networks like this really help things happen. So participate, be outgoing, do what you can. And finally, stay safe. 2020 is a strange year. Thanks.
Well, thank you so much. Really, thank you for the youngers like me that are starting. That is very motivated to do to listen that. So thank you for my part, and I suppose that for everyone. So now, Sergio, if you want to say something.
Yeah, just to thank you, James, again. It's really inspiring. That's the idea of these talks, to inspire new people. Yeah, to start to work exactly as you said. Yeah, just inspiring new people to start to work and not give up at the beginning, because we know that's really hard, especially
Yeah.
in the beginning.
And I was a bad student. So like bad students don't give up either, you know, like
Exactly.
don't do it.
But it's really, really important. We received a lot of questions, really nice questions. We had a lot of attention.
Yeah, good questions. It was I enjoyed it.
Yeah. So
Well, thank you so much.
No worries.
Now,
thank you, guys. Take care. See you soon.
Too.
Yeah, we hope the same.
Yeah.
All right. Cheers. Thank you.
Ahora vamos a caminhar con conocer de nuestro evento, ¿no? De la CNIda, lo primero evento que existe durante estos días, más de una semana, con todo este. Tenemos una palabra de lo gran fundador de la CNIda, de la network, de la red, que van a hablar ahora para nosotros, que nos brindan con sus palabras, es Fabián Acuña, de Argentina. Fabián.
¿Ahí me ven?
Sí, mucho gusto de tener usted acá.
Bueno, igualmente. ¿Ya puedo comenzar?
Sí, la palabra es tuya.
Bueno. Gracias, Sergio. Bueno, unas palabras muy breves, porque bueno, ya ha pasado bastante tiempo. Y simplemente quería comentarles que en estos días estuve viendo las exposiciones, ¿no? De estas de 20 minutos que hicieron muchos de ustedes. Y también pude ver ayer, sobre todo, estas primeras charlas de 3 minutos que dieron los jóvenes investigadores. Contanos tu investigación en 3 minutos, ¿no es cierto? Y realmente he quedado muy gratamente sorprendido por la calidad de las charlas, la variedad. A mí, yo hace 30 años que estoy en la profesión y he tenido la suerte de conocer gente nueva que no sabía que existía. Y creo que esto es un poco uno de los objetivos de la red de la CNIda, que se ha cumplido no perfectamente, justo un año después de la creación de la red, cuando comenzamos a discutir en el laboratorio con Agustín y otros colegas la realización de la red en el COLACMAR. Éramos un poco, no digo escépticos, pero sí dudosos de la respuesta que íbamos a tener. La reunión que se hizo en Mar del Plata éramos 30, con suerte, y quedamos muy contentos. Hoy intercambié un par de mensajes con Agustín y resulta que ahora la red somos 150, ¿no? Entonces esto es un dato más que alentador, ¿no? Que 150 personas de Latinoamérica se hayan sumado a la red es realmente un dato muy, pero muy positivo, y más aún teniendo en cuenta las dificultades que habitualmente tenemos en nuestros países, ¿no? Con pocos recursos, con poco apoyo a la ciencia, sobre todo a la ciencia básica, ¿no? Lo que decía James, ¿qué son los cnidarios o para qué sirven o no vas a tener trabajo? Creo que esto todos lo hemos vivido en algún momento, pero bueno, esto demuestra que en Latinoamérica hay mucha gente trabajando en muchos temas diversos y además con buen nivel de datos científicos, ¿no? De investigaciones científicas que muchas veces se plasman en publicaciones muy buenas, ¿no? Que son reconocidas y citadas en todo el mundo. Entonces, realmente he quedado más que contento por esta iniciativa de ustedes, particularmente la gente joven, de realizar estas actividades virtuales a un año de la creación de la red. Otro de los objetivos de la red, que nos planteamos allá acá en Mar del Plata, en realidad, fue la realización de un tercer simposio que se iba a hacer en el COLACMAR. Lamentablemente, por la pandemia, el COLACMAR que se iba a hacer en Panamá, ha sido postergado un año más, para el año 2022, pero esto de ninguna manera podemos tomarlo como un obstáculo, ¿no? Sino al contrario, como los latinoamericanos estamos acostumbrados a saltar obstáculos constantemente, y quizás eso es el motivo que muchas veces nos impulsa a esforzarnos más, ¿no? Entonces, bueno, yo quería invitarlos a todos a seguir conectados en la red, difundir la red, tratar de conseguir más gente. Hay mucha más gente que seguramente se podría sumar. Por ejemplo, no veo en la red mucha gente que estudie corales, ¿no? Aunque entiendo que la gente que estudia corales tiene sus propias asociaciones, ¿no? En México, por ejemplo, o a nivel internacional también. Pero también estaría bueno que se sumaran a esta red. Y también tener en cuenta que la red depende de todos, ¿eh? La red no la hace un grupito de personas que se juntó una vez y que, bueno, dieron forma a esta red y que se van a ocupar siempre de esta red, ¿no? De alguna manera, los 150 que estamos en la red, tenemos que sentirnos de alguna manera responsables, ¿no? De mantener esa red y tener en cuenta que las personas, sobre todo las que ahora forman la junta directiva, no van a estar siempre, ¿no? Y en algún momento será necesario un cambio de personas, de caras, que es doblemente positivo, ¿no? Por un lado, cuando las mismas personas están siempre con los mismos cargos o en las mismas comisiones, se terminan cansando. Y por otro lado, eso, bueno, eso es algo que no debe pasar, y una renovación cada tanto va a ser más que bienvenida para evitar esto por un lado, y por otro lado, gente nueva trae nuevas ideas también, ¿no? Lo cual es sumamente positivo y necesario para la red. Así que bueno, por un lado, ya para terminar, primero agradecimiento a todos los que han colaborado con la red, particularmente ahora a esta junta, esta comisión organizadora de estas actividades virtuales, que lo han hecho muy pero muy bien. Y bueno, mis felicitaciones, ¿no? Porque realmente venimos a decir, gratamente sorprendido por todo lo que han hecho. Ha sido todo muy, pero muy bueno. Y bueno, simplemente darles un gran abrazo fraterno, latinoamericano, y bueno, les pido que sigamos en contacto, ¿no? Muchas gracias.
Muchas gracias, Fabián. Bueno, chegamos ao final, agora, sim, nosso cierre definitivo. Ainda temos alguns vídeos de los 3 minutos que vão ao ar nos próximos dias, pero as atividades em vivo já se foi, sim? E agradecemos, agora em português, agradecemos a todos que estiveram aqui, que ficaram assistindo as nossas transmissões. Foi um momento muito importante, como o Fabián mesmo disse, é importante que vocês se comuniquem com a rede, se precisarem de alguma coisa, se tiverem interesse em alguma coisa, nós vamos tentar agilizar da forma mais aberta e disponível possível, né? Que tenha essa interação entre as pessoas dentro da rede, né? Para que isso seja viável e que a rede cresça cada dia mais, né? Então a gente já tem um número bastante grande de pessoas associadas e que estão integradas nas discussões, mas a ideia é que isso seja uma rede completamente aberta e que todo mundo que tem algum interesse em Cnidaria tenha acesso a essa informação e que a gente consiga ajudar de alguma maneira, né? Então a ideia da rede é realmente ajudar. Então é isso que a gente deixa como um recado, e até a próxima da nossa reunião, né? Pilar?
Bueno, no sé si yo hablo de mi parte. Bueno, muchas gracias a todos también por participar, a todos los científicos y a todas las personas que también son iniciantes en la ciencia por participar. También agradecerles a todos los que están mirando, sean o no parte del estudio de cnidarios o estén trabajando en ello. Felicitaciones a mis compañeros por la organización, la verdad es que salió excelente. Y también gracias por permitirnos a los más nuevos que estamos arrancando participar en estas actividades, y también lo mismo que decía Sergio, que espero desde mi punto personal, igual que el de todos, que cada vez se sume más gente a participar en la red, jóvenes, grandes, interesados, más desde el lado por ahí como amateur, siempre son bienvenidas y cualquier duda que tengan les vamos a estar contestando. Esto sirve mucho para conocernos entre todos y saber tal vez de un trabajo que uno imagina o quiere hacer y probablemente haya otra persona en el mundo que lo esté trabajando y nos pueda dar una mano o aunque sea decirnos que nos contactemos con otra persona, así que también por eso los invitamos a que sean parte de esta red, que trabajamos muy, muy bien en equipo, o eso por lo menos parece. Así que gracias y gracias a todos por participar.
Muchas gracias a todos. Ahora vamos encerrando la
Bueno, y
transmisión.
gracias James
¿Sí?
también. Thank you, James.
Thank you,
for being
James.
here.
Thank you.
Thanks to all of you, and thanks for everyone for the questions.
Yeah, it's very nice. So, see you.
Gracias, Sergio, por todo. Gracias, Pili.
No, por favor.
Un abrazo.
Abrazo.